This is a video review of Module 2 Quiz Part A, and I'll review a number of questions that are most missed. First question is number 5, which asks the zone of a developing long bone that is a template for bone spicules, and where is that created? Well, the template is actually the zone of calcified cartilage, not the zone of resorption and bone deposition. The zone of resorption bone deposition is where the bone, the osteoblasts, are depositing bone matrix upon the calcified cartilage template. The next question, number 6, which is a true statement about the epiphysis plate of a long bone, developing bone? Well, it lies between the epiphysis and diaphysis. Remember, the epiphysis is the end. There are two epiphysi in both in a long bone, one at the distal end and one at the proximal. And so the epiphyseal plate actually lies between one of those ends and the main shaft of the bone, which is called the diaphysis. And number 8, the zone of calcification in the epiphyseal plate is mainly responsible for the growth of the length of a long bone. What that is false. The zone of proliferation, where the cartilage is dividing and all in one direction, forming stacks of chondrocytes, is the location of the growth of the long bone. And number 13, this is all about which of the following is the potency of a cell that can differentiate into any of the more than 200 cell types in the body. Well, that is totopotent. That is totally potent. Pluripotent means multiple or many, multi or several, oligo or few, and uni is just differentiating into one cell type. Okay, so number 16, an astrocyte functions like a macrophage in the central nervous system. Well, no, astrocyte is actually supporting the neuron between the capillaries of blood vessels and the neuron, cell body mainly, but it's the microglia that are behaving like a macrophage. So review those in the nerve lecture. And let's see, the next one, number 18. Canaliculi in bone contain processes of osteoclast. No, they contain processes of osteocytes. So review that. And number 19, collagen types and where they're found. Well, osteoblasts secrete which type of collagen? That's type 1. And chondroblasts and chondrocytes that may be forming within the body of the cartilage will produce type 2. And type 3 is in reticular fibers, and type 4 is found in the basement membrane, etc. So review those collagen types and where they're found and which cells make them. So number 20, the eye band in skeletal muscle is primarily composed of myosin filaments. No, the eye band is composed of actin filaments. Remember, the actin filaments in the eye band are anchored in the Z line or Z disc. They extend and interpose between myosin filaments in the A band on one side of a sarcomere, and then on the other side the same is true. So some of you need to review that relationship between myosin, actin, and the banding in skeletal muscle. And so number 21, recognizing intramembranous versus endochondral ossification. If you see any cartilage present, which you don't see here, you see the eosinophilic bone matrix. You see the pattern here with no cartilage around, very red. Cartilage would have been bluish staining, and you would see a whole different pattern here. You see the osteoblasts on the surface here. So this is a bone spicule, and another one here. So this is occurring within a membrane, intramembranous ossification. Okay, and number 22, recognizing sensory ganglion cells. Okay, well, of the cell types present here, or listed, Certainly not smooth muscle cells, although a smooth muscle cell has a nucleus in the center, but does not exist with the satellite cells surrounding it. And you also do not find nuclei, that is nuclei, in a smooth muscle cell, and the cytoplasm would be much more eosinophilic. This is slightly basophilic in the very large round cells. So these are sensory ganglion cells. So review the nerve cell types and distinction between those and other cell types. Okay, and number 23, parts of a nerve fiber. Well, here we ask you, we said the nucleus indicated by the arrow in the high magnification belongs to which of the following? Okay, here's a nucleus. You can see a wrapping here. You see a clear space, and you see kind of a dot in the center as compared also in these profiles. Well, that's the actual axis cylinder of the axon, and this clear space is the myelin, and this nucleus belongs to the Schwann cell. So you need to review that, some of you. Okay, number 24, the wrapping of skeletal muscle. Remember, the outermost wrapping is the epimysium that surrounds a whole group of muscle fascicles, and then whole groups of muscle cells which form a fascicle surrounded by perineurium, and then individual muscle cells that you can see here surrounded by a very delicate connective tissue that is stained blue here, and that's endomysium. Okay, so question 25, recognition of white blood cells. This is a neutrophil. And the question asks you to choose the percentage in a white blood cell differential count. That is, what fraction of the count would be this type of cell. First, you have to recognize the cell, so you need to review all the white blood cells, lymphocytes, etc., and be sure you know the percentage range. Number 26, recognition of white blood cell like a monocyte. And then what does it do? Well, in the circulating blood, it just circulates. But it can become a macrophage by going out of the vascular system, which it does in response to some stimulus, some foreign substance. And number 27, the structures indicated provide channels for communication. Well, you need to recognize that this is bone tissue, and these are canaliculi, and these large oval structures are lacunae containing osteocytes, and the processes of osteocytes extend through these tiny canals called canaliculi. And so number 28, okay, so these are osteoclasts because they're large cells. They're located on the surface of bone, and they contain multiple nuclei. Now, you could get this confused with megakaryocytes because they're present also in relationship to bone and the bone marrow, but megakaryocytes only have one huge nucleus. Remember, it's a very large cell like this one is, and the nucleus is so large because the cell's so large it has many times the normal amount of DNA in the nucleus, polyploidal. Okay, number 30. Okay, the cells in the epiphyseal plate within the yellow circle are active in which of the following ways? Well, you're looking at this from left to right, you see stacks of cells here. That's the zone of proliferation. You see the cells are larger here, and this is the zone of hypertrophy. And then you see here the deposition, the calcification of the cartilage. And then later on, the deposition of bone is exhibited by the osteoid, the little pink layers on the calcified cartilage. So in this area here, cell division has stopped. So there's no growth going on here. It's just enlarging and getting ready for the matrix to calcify so that bone can be deposited. Okay, number 31, recognizing the hyaline cartilage and the collagen type 2 in its matrix. Okay, so you need to recognize this as cartilage, and also recognize that in cartilage the chondrocytes and the chondroblasts produce type 2 collagen. And let's see, number 32. What type of cell makes up the layer that surrounds the three structures indicated by the arrows? Well, observe the enlargement on the right. And so you see the tissue between the dotted lines. So this is a fascicle, and you know that because you see all these multiple nerve fibers in cross-section. So this is wrapping a bundle of nerve fibers, a single fascicle. So what wraps a fascicle? Well, that's perineurium. And the perineurium is composed, we gave multiple possible answers. Fibroblast, type-like cell. So we were pretty lenient on this, perineurial cell, etc. But if you didn't pick one of those, then you got it incorrect. So review that. And question 33, recognizing the features of a basophilic erythroblast, are all the stages where you have a blast cell here, and then this is a basophilic erythroblast where you have a very large nucleus and a rim of very intense cytoplasm that's composed of ribosomes that are making hemoglobin. So the correct answer to that was cell C. And question 34, osteocytes make type 1 collagen. So we wanted you to answer, first of all, you have to recognize this is bone. From the spicule here and the cells here and the canaliculi, you can see, and this is Mason's trichrome, so this is collagen here stained blue, and these are osteoblasts that are forming bone matrix. And so they produce type 1 collagen. This is type 1 collagen here. And the last question, we said, okay, you have two views. You have a low power of skeletal muscle, and you have a high power stained with iron hematoxylin. So, in this, we pointed to a Z line here and a Z line here, and the question said, what is the name of the functional unit that lies between two consecutive dense lines, indicated by the arrows? And that has to be a sarcomere. And the key word here is, to reading the question carefully, is consecutive. We just pointed out a couple of them, but we wanted you to think about that and realize that two Z lines in consecutive line define a sarcomere. Okay, so that concludes Module 2, Quiz Part A.